Hi, this is Mr. Wartsky again. Today we're going to cover the second screencast of recombinant DNA technology. This will be the first slide of the second screencast. If you have any questions, please rewatch the video over again, read your textbook, or ask me questions. So radioactive probes can be used to find the location of DNA. The radioactive isotope that's attached to a deoxynucleotide. DNA synthesis will put the isotope and the deoxynucleotide in place and allows us a way to find DNA without staining it. Now today we can use fluorescent tags on deoxynucleotides and nucleotides, and the DNA, when a special light or laser is shined on it, each nucleotide will give its own glow at a specific color. So you actually know which deoxynucleotide you're looking at. DNA sequencing is another technique that we can use, and there's DNA sequencing with dideoxynucleotides. Man, that was rough. Dideoxynucleotides, deoxynucleotides, which means that it is missing both the hydroxyl groups on the 2 and 3 prime carbon. So right here at the bottom is a dideoxynucleotide, where this is a deoxynucleotide. Dideoxynucleotides are referred to as chain terminators because when you put a dideoxy in its place, there is no 3 prime hydroxyl group right here to add the DNA on. There's no 3 prime right there. DNA synthesis will stop. And oftentimes dideoxynucleotides are shown with this little asterisk to show that DNA synthesis will stop. So you will have 4 different dideoxynucleotides: DDATP, DDCTP, DDGTP, and DDTTP. And they will stop DNA synthesis cold. So you will have 4 different wells. Each well will be a different dideoxynucleotide with regular nucleotides too. You run the gels and the DNA will separate. Right? So we have the A, the C, the G, and the T wells. In this one well, you will have dideoxy ATP and deoxy ATP. All the other nucleotides, you'll have DNA polymerase and primers. This one, you will have just all the nucleotides, but you will also have dideoxy CTP in the C well. You'll also have primers and DNA polymerase. In this well, the G well, you'll have all the nucleotides and dideoxy GTP. Of course, you'll have polymerase as well as the primer. And the last one, you'll have the nucleotides, and you'll also have dideoxy TTP, DNA place, and primers. So when you do DNA synthesis, sometimes DNA synthesis will go all the way to the end of the strand right here, and you'll have a full strand of DNA, which is fine. Sometimes you'll put down the adenine on this right here, thymine, and if you put the dideoxyadenine, DNA synthesis will stop. So that's why you have this. Now, when you run the gel, it'll be a very small fragment, so it'll run really far. The large fragment here will be at the bottom. If you take a look at the C well right here, you'll put DNA synthesis right here. It could stop at the first C, right? You have a really small, or it can stop at the third C. Was that the second C? That's second CCG. Yeah. Or it can stop all the way over here on this G here. Right? So when you run the gels, you'll have DNA sequence or DNA strands of varying lengths. So you read from the smallest to the longest would be A, right? G, T, T, G, C, T, A, C, C. So that is the complementary strand to the DNA. So then you just write the opposite to that, which would be T, C, A, C, G, A, T, G, G, and that would be the strand of DNA. Now that we do something similar, but we use fluorescent tags, and instead of running a gel, we let the fragments sink in a gel. We let gravity do its place, so the larger fragments will sink faster and farther than the smaller fragments. So if we let them sink and then we shine the light and we say that the T's are pink, the C's are blue, the yellows are G's, and what is that? Green is A's. We can shine a laser on them and just read from the larger bright big up. Now, if we do this in a tube and let gravity take over, the larger ones are going to sink faster than the smaller ones. But they ran a electrophoresis, so the smaller ones are here and the larger ones are there. So we can also do DNA fingerprinting with uh, dideoxynucleotides, um, or we can just do it with restriction enzymes. And what we can do is DNA fingerprinting. We're not really looking at the DNA itself, but we're looking at chromosome size. So we take the chromosomes and we cut it with restriction enzymes. We put it in a well, we run the well or run the gel, and we will see DNA of different sizes moving at different distances. We call this restriction fragment length polymorphisms or RIFLPs, RFLPs. So we will take homologous chromosomes because that's one of the controls, right? Because homologous chromosomes should be similar to each other. And some of the DNA sequences are going to be slightly different as polymorphisms. So the restriction enzymes will probably cut at different places on the chromosomes. So you'll end up with different chromosome sizes. Now, we used to do this with 7 chromosomes, with each with a different restriction enzyme. So you end up with different size chromosome lengths, and then you ran and then you compared. That's old school. Right? So this is what we used to do. We used to take the DNA, cut it, and then ran a gel. And then we used to compare down here. So obviously the sample is plant A because the bands match up. The new way, what we're looking at are the STRs. Remember those small tandem repeats? Yep, they're found on different chromosomes, like chromosome 1, 2. There's 3 of them on chromosome 4, all the way down. We have one on X and Y, because in case you want to know the gender of the person that's involved in the crime. Right? So what we're going to do is you're going to take a look. Now, these STRs are of different sizes in different people. You're going to amplify 7 or 8 of these things through PCR and take the mass, and you'll have a mass, right? And then you'll have an STR on a Y chromosome, because if you have a Y, then you're a male. If you don't have the Y, then you're a female individual. So then you'll take a look at the mass. So these are the STRs at the top right here, and this is the mass. It's like a little bar graph. And all you do is compare the size of the STRs of the sample to your suspects. And if they match up, then you have a match. Nucleic acid hybridization is also another tool or technique in which we compare the DNA of 2 different sources. So what we'll do is take DNA from 2 sources, heat them up, the hydrogen bonds break apart, we cool it down, and the DNA will bind. Now, sometimes the DNA of the same species will bind together, that's fine, but sometimes DNA from the different species will bind together. And when they bind together, that's where they are similar, and when they don't bind, they form these little bubbles, that's where they're different. And we can use comparisons by comparing how many bubbles there are to tell you how similar the DNA is of the different species. We can also use it to find genes that are similar between species. So, for example, fruit flies have an enzyme called alcohol dehydrogenase, and we have an enzyme called alcohol dehydrogenase. If we know the location of the fruit fly enzyme and we cut that DNA out, we can do nucleic acid hybridization to find the location of the same gene in the human genome. Now, if we tag that fruit fly gene with a fluorescent tag or a radioactive probe, it makes it finding it very easy. And this is a picture of nucleic acid hybridization. So where they don't bind, they are not similar. Where they do bind, they are similar. Sometimes if they don't bind at all, well, then they are not similar at all. There is another tool out there which is brand new called CRISPR, and it's used with the enzyme Cas9. So CRISPR stands for clustered regularly interspersed short palindromic repeats. And Cas is a CRISPR associated gene, and that's basically from the Streptococcus pyogenes, and that's what actually cuts the DNA. This was first found in archaebacteria, then eubacteria, and what it does inside the bacteria, it will get rid of the viral DNA from bacteriophages that have inserted into the bacterial DNA. They're in prokaryotes, they have these segments that are short repetitive bases. And what it will do is look for those bases, cut out the DNA between those bases, and that's usually where the bacteriophage DNA ends up. So what we do now, well, the protein CRISPR uses an RNA sequence that binds to the repetitive bases. And on the protein, I should say protein, is an enzyme called Cas9, and that's what cuts the DNA. What we can do is we can take our own RNA sequence and attach it to the CRISPR so that it can find the DNA that we want. And when we cut out the DNA, then we can either put another DNA in, or we can remove genes that we don't want there. Now, this is telling you how CRISPR works by cutting out the viral DNA. Now, this is the second defense against for prokaryotes that we talked about against viruses. The first were the restriction enzymes. So this what tells you what we do. We add a synthesized RNA sequence, right? Find the section of DNA that we want. We can cut them out. We can add our own genes. We can do that. Now there is another one, another enzyme, CPF1, that can cut in a sticky end fashion. So it's not blunt end cuts, just like Cas9. And here's what CRISPR can do. Right, we can cut out and add. We can repair. Or we can repress or activate the DNA. So what else can we do with CRISPR? Well, we can cut out bad genes and turn them off. Right? We can also have the CRISPR act as a repressor to stop DNA synthesis. We can insert good genes. We can add a transcription factor to CRISPR so that we can turn genes on. We can add fluorescent tags to CRISPR to find the genes. We can even block RNA polymerase to turn genes off. Is that it for CRISPR? No. We have now been able to add methyl groups as well as acetyl groups to either stop protein synthesis or increase protein synthesis within a genome. And we're going to stop there and finish this in the next screencast. Have a good night.